Okay, today we're going to be talking about coronavirus encore, since it seems to be the health scientific problem of the day. And we're going to talk about clinical trials because it's very pertinent in that they have just announced yesterday that they're starting phase 3 clinical trials for 2 vaccines in the United States. So I thought it'd be a good idea to just go over this whole area. And like everything else in life, and particularly in science and medicine, you can get lost for the forest by the trees, and so this is an attempt on my part to explain things in enough detail so that you can understand what the situation is without having to get into all of the arcane little details of the thing. The first is, what is a clinical trial? Well, a clinical trial is an experiment done on human beings. And by definition of what an experiment is, that means that we don't know what's going to happen when we do this experiment. And that's one of the problems. So things have been worked out over the centuries, really, in order to do this so that hopefully the volunteers for this clinical trial won't be adversely affected by the whole thing. Like everything else, I like to go back into the history of this whole business, and it's fortuitous that really at least the first clinical trial that I'm aware of was in immunology and was the vaccine trial by Edward Jenner, published by him back in 1798. So that's 222 years ago. It's an interesting study because what he did was he published a monograph in 1798, and in 1796 he had submitted a paper to the Royal Society in London, which was really the only place that you could publish back in those days, unless you published it yourself and paid the printer and so forth to get your thing published, which is what he did. In 1796, he sent to the Royal Society sort of a manuscript that described his experience with 10 people who had previous episodes of cowpox infections, and usually these were the milkmaids in the countryside. A lot of dairy cattle. It was just west of London and into around Oxford. And what he showed in this first 10 series was these were just simply observations. And he observed that if the history of the person was that they'd had cowpox previously, when he inoculated them with smallpox, they didn't get a take. They didn't suffer any ill effects, and nothing really happened. And so he submitted this. But the Royal Society didn't think this was all that significant, and so they basically sent it back to him and rejected it. And so he went back to the drawing board, and he started actively taking patients with that had not had cowpox infection. Sorry about that. And so what he did was that he accumulated more cases. And one of the first persons that he inoculated with the cowpox and then challenged that person with smallpox was a young boy, 8 years old in the community. And so when he challenged the small boy with smallpox, nothing happened. And so then he worried that he didn't have a really live virulent smallpox injection. So he did it again, and which I thought was pretty brave to do. And again, nothing happened. And then he went on and he had other cases, and he put them together with 25 or 26 cases into another report that he sent off to the Royal Society. Again, the Royal Society did not accept it. And so he went to a publisher in London and paid to get it published. And that was the beginning of the cowpox or the smallpox vaccine that was really the foundation of immunology and still is. So the thing is, the question is, was that an ethical thing to do? Should he have done that to this poor little boy that lived down the street? And so the ethics of vaccination and then testing as to whether the vaccine worked or not is the question that we're going to be dealing with today. And the next experiments that were done in vaccine work and so forth that are of note were the experiments that Jonas Salk did in 1955 with polio and polio vaccines. And just to recapitulate a little bit of what we've talked about before, Louis Pasteur had done a series of experiments or experiments back in the late 19th century or in the 1880s, where he had experimented with animals, domestic animals, first with chickens and then with sheep and cows, first with what they called chicken cholera, which was a neurological problem that the chickens would get when they got infected with a bacteria, and then later on with anthrax. And I think these experiments are important because of the fact that by confining the whole thing to animals, to domestic animals, you could do very clean experiments. And so I think the best example of that is the anthrax. And the anthrax experiment became really celebrated because Louis Pasteur had announced to the French Academy of Sciences that he had this great vaccine. And the veterinarians for anthrax that had infected sheep and cattle, and the veterinarians were a little bit skeptical. And so they challenged him to an experimental demonstration of the efficacy of this vaccine. And they went a little southwest of Paris into a little village that was near Fontainebleau, and they segregated off 50 sheep, of which 25 Louis Pasteur vaccinated, and then 50 sheep were the controls. And this is really important, and we still do this today. He didn't give the control sheep a placebo injection. They just remained unvaccinated. And then they got a booster shot, and then 2 weeks later, and then a month later, Louis Pasteur came back and challenged all 50 of the sheep. And within 2 or 3 days, with live anthrax, and within 2 or 3 days, the control animals were dead or dying, whereas the vaccinated animals were just, good as new. Now, and that shows the power of the experiment when you're using experimental animals. Now, the rub is, it's difficult to do that nowadays with humans. So Jenner did it in 1798 to a little boy down the street, and but now we can't necessarily do that today, except, and this becomes the ethical problem that we're dealing with. If you're dealing with a pandemic where thousands of people are becoming infected and thousands of people are dying, the question is, should we do a challenge kind of a trial where we take not a huge number of people, but enough to be very, very confident if the results are clear cut, like with Louis Pasteur and the sheep and the anthrax? Should we do that? Or should we do what is now considered the only way to go, really, because it's the only ethical thing to do, which is to do a clinical trial? So I started with Jonas Salk and what he did in 1955 with polio. And you have to remember that polio was really that was scary times. And he had generated a vaccine, and he had vaccinated experimental animals and monkeys and so forth, and then found that his vaccine was working. And he then went to a school that was, he was in Pittsburgh, the University of Pittsburgh. He went to a school that had crippled children there, and they were crippled for the most part because they'd already had polio. And he went ahead and inoculated these crippled children with his vaccine, and then he measured then antibody levels and so forth to see whether there was an immune response. And on the basis of this trial, and then he did one other trial, it looked like things were a go, and the March of Dimes, which was a private philanthropy, was backing him money-wise. So they started a huge inoculation program all over the country. They never really did do a clinical trial the way we do clinical trials today in polio. But I was only 12 years old in 1955, and so I was very susceptible. And my parents were scared to death of polio, so I can remember what that was like. So nowadays we do clinical trials to test drugs or vaccines, and those are called a interventional trial, because we intervene and we do something to the participants of the trial. Or you can have a observational trial, so that you take people and you put them on a low carb diet, and you put other people on a low fat diet, and then you follow them along and take measurements and so forth over time, and to try to find out which diet is the best. So there are basically 5 phases to a clinical trial. So there's this phase 0 is the first, and then there's 1, 2, 3, and 4. And phase 0 is really a pharmacological study in humans to find out what happens when you vaccinate the humans for the very, very first time. And there's 2 basic things that you study in this, and you don't do too many people in this. What you're trying to find out is what the vaccine does to the person. That's called pharmacodynamics. And the other thing you want to find out is what the person does to the vaccine. And that's called pharmacokinetics. And actually, when I did my first clinical trials with interleukin-2, that's what I did. I did pharmacodynamics and pharmacokinetic studies to try to make sure that the dose was in the range that I wanted it, and also that I didn't do anything terribly wrong to these people. So that's phase 0. They don't talk about that, but that's phase 0. Then phase 1 is to try to find out, the purpose of phase 1 is to try to find out a safe dose, and also sort of in a range that you think that it seems like a reasonable dose. And so what you do in a phase 1 trial is, and you don't do too many people in a phase 1 trial. It's less than 100, and it's usually 20 or 30. What you do, if you've got things bracketed correctly, what you do is you take 4 people at a time, and you give them what you think is a low dose. And what you can think that based on animal experiments that have been done prior to this time. And so what you need to do before you can get approval to do your phase 1 trial, you need to show the data for toxicology in animals, small animals, and then bigger animals like dogs, small being rats and mice and guinea pigs and so forth. And then things like dogs, and then primates, and small monkeys and large monkeys. And in all those experiments, you see you can ascertain what the dose range ought to be. And so in your first clinical trials in phase 1, you calculate a dose that you think is going to be below what you found from your animal experiments. You take 4 people, they get that dose, and then you measure pharmacodynamics and you measure pharmacokinetics in those people. And for the most part, if you've bracketed the dose range properly, they won't suffer any ill effects. And that being the case, then you go on to the next 4 people, and you dose increment. You can either add 50% or you can double the dose or whatever, and it depends on how you do that whole thing. And so then you move along that way. And ultimately you get to a dose whereby you start to see clinical symptoms, signs and symptoms, some of which you may think are okay in terms of what you anticipate, and some of which are unanticipated, and therefore those are the ones we call side effects. And so if everything works out well, as you do 4 people and then 4 people and then 4 people and so forth, you end up with what is the maximum tolerable dose. And so then what you do is you back off from that maximum tolerable dose to arrive at a dose that is high enough to develop an immune response, in this case because it's a vaccine. And that becomes the dose. Or sometimes you get 2 doses, a lower dose, and a little bit bigger dose before you move on to the phase 3 trials. And so that in these dose finding safety studies, you don't need too many people in those kinds of studies. So then you move on to the first efficacy trials in phase 2. And then you need more patients. You need more than 100, somewhere between 100 and 1,000. And depending on the kind of an experiment you're doing, usually with vaccines and what they're going to be doing with coronavirus is they're going to have a control group that doesn't get the vaccine. And then they're going to have a group that does get the vaccine, and the dose that you've established in your phase 1 trials, your dose-finding safety studies. And then you use moderate numbers of people, somewhere around more than 100. And what you're looking for in this instance is a sort of proof of concept that the vaccine works. And in this instance, there's basically the real way that they do it nowadays in immunology is that they test for immune responses, and so they look for antibody responses that appear in the blood plasma, and they can also take out lymphocytes and test for T cell responses and so forth. And they can also measure cytokines that are produced as a consequence of the immune reaction. And if they get, and that's what's happened already in the coronavirus story with some of these vaccines that have been brought up through the system. They know, and this is what they've been having press releases about and so forth. They know that their vaccine has resulted in immune responses that are measurable, and they're in the range high enough so that they would anticipate that they would hopefully be protective of an infection with the live virus. So that would establish then the fact is that this dose of vaccine is safe, it works. And now it works in terms of generating an immune response. And now you have to go on to phase 3. And this phase 3 takes many more patients, or subjects, I should say. And that's a critical thing in clinical trial jargon and so forth. It's you don't want the person who is volunteered for this trial to get the idea that their doctor is treating them, because this is an experiment. So you don't want to call them a patient. You should be calling them subjects or volunteers. So in order to do a phased trial, what they've announced just yesterday is that they're for each vaccine, and there are 2 of them that have started now, in clinical trials now in the United States. They're planning on 30,000 people. And those will be divided into those that get the vaccine and others that serve as controls. And they'll probably get a placebo, which would be a masked vaccine. It's not a real vaccine. It's usually the liquid that the vaccine is suspended in. And so here comes the conundrum in this whole business, because now what do? Well, the reason that they've calculated that they need 30,000 people, that's called the sample size, and that's based on statistical analysis of the anticipated results. How many of them do they want to become positive in their experiment versus the control group, or how many patients, well, I should back up a little bit. And so what they do, and the reason it takes so many people, is that they have to let them live their lives. And they try to pick people and areas of the country and so forth where there's a fair amount of virus. The more the better, because the next thing that it takes a while for everybody to become exposed and either come down with symptoms and get sick and so forth. And that's going to be the proof in the pudding as to whether or not the vaccine works. And so they've calculated evidently that you need 30,000 people, because they're not looking for too many of these vaccinated people to be exposed. And that's based on the prevalence of the virus in the community. So if out of 30,000, only 3,000 actually become exposed, and then of those 3,000, let's say only 2,000 actually became infected, even though they were exposed. And then out of those 2,000, in the control group, maybe you'd hope that 2,000 of those people would get sick. Because that's the control group. And then in the vaccinated group, you would hope that nobody got sick, or nobody became positive in terms of tests for the virus or whatever else. But it doesn't work out that way usually, because there's a certain efficacy and when they talk about, well, it's 60% effective, that means that it wasn't in a vaccinated group. Some people, even though they were vaccinated, still got infected and still got sick. And where if it's 100% effective, then you wouldn't have anybody. And so so that's phase 3. And on the basis of the phase 3 trials, which it depends on the infection. Now that's going to take some months. And this is the reason sometimes it takes years to get enough people inoculated and enough people infected in order to have the results so that you can then get this vaccine approved by the authorities. And in the United States, the authorities are the Food and Drug Administration. If the drug is approved, then it's out there and it's released, the vaccine, and then they do phase 4. The companies that are marketing these agents, and phase 4 is called a post-marketing analysis. And so what they do is to the people that have then received the drug, they track people and look for side effects that are very rare but still real, They want to know what the whole gamut of the administration of this drug actually is. And sometimes, in the phase 4 data that have come out, they've removed drugs from the market because the side effects have outweighed the risk of the side effects have outweighed the benefit from the drug. So that brings me to what I started with in this discussion, and that is, and I've thought about this a fair amount, and I think that the ethics on this are with those people, and there have been papers out in the best journals in the last couple of months by the best people in the business, epidemiologists and ethicists, that are saying, this is an emergency. This is a pandemic. People are dying all over the world. We can't wait for a clinical trial. And in fact, we should do a controlled experiment with, 50, 100 volunteers, and have a vaccinated group and a control group that we then know that we infect those people with. And then we get data. And it doesn't take forever. And we know whether this is going to work or not. And I'm with these people. I think it's crazy that we're doing these huge, great 30,000. It's each vaccine that they're going to call for 30,000 volunteers, at least in the United States. Meanwhile, in China, I read reports just in the press that they're already doing the challenge. They're calling for volunteers. They're using their military recruits, and they're also calling for volunteers to come into trials that they're going to be vaccinating them, and then they're going to be challenging with. And so, what's going to happen? China's going to have a vaccine before the good old United States of America. And I'll stop there.